Bismillahirrahmanirrahim. This is another video on some of the questions that you all have been asking me. So this contains nearly all the difficult MCQs which are possibly can come again in the exam, and or something which is sort of related to the question can also come. So I hope this helps you all. This is a little difficult question number 6. Now, the diagram shows a dialysis machine, a dialysis fluid out, dialysis fluid in. So this is the blood from the patient going in here, and then it is coming out here and going back into the patient. So basically, what is a dialysis machine for? It is to remove the urea. So it is to remove the urea because the kidneys have failed. And what do the kidneys do? They remove urea, they remove not excess urea, they remove urea and they remove excess salts and excess water. So in a person whose health problems affect only the kidneys, which substance will move as shown at P and Q? Now P, the dialysis fluid, something is moving into the dialysis fluid. And what is moving out of the dialysis fluid? This Q has to be nothing but urea. So Q has to be urea, so it can be either be this or this. Now what do we have? We have glucose moving into it. Why? Dialysis fluid does have some glucose in it, but it is usually lesser than what is in the blood. So dialysis, it's not going to diffuse out, but it's nearly the same concentration. So there's not going to be any. So if you've got glucose 100 in the dialysis fluid, in the blood also is nearly 100. It can be sometimes more. So it is usually the dialysis fluid contains glucose. Basically, it is not the removal of glucose that is needed. So what we have to understand is the answer is D, but this is actually what we have to understand is that in a dialysis machine, urea is going to be moving out of the blood. This is the blood of the patient, which I am coloring in yellow. This is the blood of the patient, and the blood is going in, and what do we have outside it? We have the dialysis fluid. So this is the dialysis fluid outside, and here and here and here. The dialysis fluid has the same concentration as, nearly the same concentration as the plasma. Now what you have to realize is proteins are large molecules. They cannot pass out of the blood. They are inside the blood, and they cannot pass out of the blood into the dialysis fluid because the membrane of the dialysis membrane, which is which is sort of a permeable, partially permeable membrane, does not allow large molecules to pass out. Another question which is challenging: in what way is the pupil reflex different from some other reflexes? Now when you say pupil reflex is different, then the others must be correct for the other reflexes. Now the possible choices are: a change in environment causes impulses in the sensory neuron. Yes, if you touch something hot, then sensory neuron carries that impulse to the brain and you withdraw your hand immediately. That does happen. A change in the environment, like you enter a room, it's very hot, all this information goes to the brain through sensory neurons which are present in your skin. Right? The function of the reflex is protective. You touch something hot and you withdraw your hand. Yes, it is a protective mechanism. The relay neurons are in the brain. Well, that's the only thing which is, you see, normally the relay neurons are in your spinal cord. If you touch something hot, the relay neuron is in the spinal cord. So, but here, now what is the pupil reflex is different because it's the eye and the brain. It's not going to from your eye, it can't go to the spinal cord and then back to the brain and then. So that would be weird. Your eyes are in your socket, your eye sockets, and you are very next to the brain. So what we have to understand is this is the only thing which is different. Now the response occurs quickly. This happens in all the other reflexes. You touch something hot and you immediately withdraw your hand. You don't wait for your hand to get burnt. So the response occurs quickly. These are, of course, all these three are correct for the other reflexes as well. But this one is the only one which is for the pupil reflex. Now question number 11, you could only get that wrong if you don't know the origin and insertion of the muscles. You see, what you've got to understand is that the biceps muscle, which is in the front, this is the biceps muscle, it is attached to the head of the humerus, while the triceps, and then it is attached to what? It is attached to the radius. This is the radius. While the triceps muscle here is attached to the ulna. So when it says what happens when the arm is straightened at the elbow, you must know where the biceps is attached. The biceps is attached to the radius, and here it is attached here and bit to the scapula as well. Here this is attached, triceps is attached here, and this is attached to the ulna here. So what you have to understand is, please look at the diagram carefully. And you can understand what happens when the arm is straightened at the elbow. D, the triceps pulls on the ulna. When your arm is flexed, so there is flexion and extension. So when you flex, you bend your arm at the elbow. When you extend this. The tricep, which is going to pull on the ulna. I mean, if somebody corrected this one, that was that you didn't know that where the triceps was attached. So you need to revise where the triceps was attached and where the biceps is attached. If you get this wrong, of course, you know when you are extending the arm, it's the triceps, but then they gave you two choices of the triceps. These were, of course, all wrong. You couldn't have got this right if you didn't know. If you're just going to do, just guess it, then of course it would be wrong. But otherwise, everybody knows it's not the biceps which contracts when you extend your arm. When the arm is straightened at the elbow, that's extension. When you bend the arm at the elbow, that's flexion. Now coming to question number 13, which statement about penicillin is correct? Penicillin. Penicillin is an antibiotic. Penicillium is a fungus which produces that chemical which we have named penicillin, and it is an antibiotic. Antibiotic means it kills or inhibits the growth of bacteria. So Penicillium bacteria, this is wrong. Penicillium is not a bacteria, it's a fungus. Penicillium bacteria can simply support and inhibit the growth of some fungi. This is rubbish. Penicillium fungal secretion can be used to treat some virus infections. No, virus cannot be. Bacteria and virus are two different things. One is an orange and one is a lemon. Virus has no cytoplasm in it. Virus has just a protein coat. That is why we are all suffering from COVID-19 and there's no treatment for it. If it was COVID was a bacterial infection, then we would have just been giving antibiotics to people. No, we don't give antibiotics to people in COVID because it's a viral infection. Then number C, the antibiotic penicillin is a waste product of some bacteria. No, it could be a sort of a product, excretory product, which is produced by the fungus, but it's not. The penicillin is not produced by bacteria. So naturally, we narrow it down to D, which is the correct answer. The fungus penicillium. The fungus penicillium. Please understand, penicillium is the fungus. Penicillin is the chemical that it produces. Produces a substance that can inhibit some bacteria. So unless you knew this very well, you would not have been able to do this question. Unless you understood very clearly that penicillium is a fungus that produces penicillin, which is a chemical, which is an antibiotic, which we have discovered that it has antibiotic properties. And of course, that is what we have now using to treat people who suffer from bacterial infections. And the bacterial infection that you study is syphilis, which is a bacterial infection. AIDS is a viral infection. Now another difficult question. The diagram shows three organisms not drawn to the same scale. Each diagram has its own scale line. Now this is a scale line. So you can see this scale line is about this length. So you have to measure this in with your ruler, then this one, and then this one. So you have to measure these scale lines. Now using the same units micrometer in each diagram, what length is the scale line for each organism? Now, if you understand what is a scale line, only then you can do this question. Okay, so now I'm going to explain to you what is a scale line. You see this diagram, there's a scale line. So this is 40 micrometer. You see this is 40 micrometer. So you would measure it with a ruler and then you will tell me how many centimeter it is and then you will convert it to 40 micrometer. Okay, here's another scale line. Now, I've given you 4 scale lines. Now this is 2 centimeter. What does this mean 2 centimeter? It means this is 20 millimeter, which means it is 20,000 micrometer. And you have to divide it by 2 because this is the 2 here. And that tells you the magnification. So 2, 1, 0, 0, 0, 0. So it's 10,000 times magnified. Whatever the diagram here might be present, could be a cell or could be a virus or could be anything. So that means this is 10,000 times magnified. Now in this situation, what will you do? 4.5 centimeter is equal to 10 nanometer. Now we have to understand is 4.5 centimeter is equal to 45 millimeter. And you divide it, so you have to convert it into nanometers. So 45 millimeter is equal to 45,000 micrometer, another 3, and it will be now nanometers. And then you will divide it by the 100, which is given here. So this is how the scale lines. Now calculate the magnification of these scale bars. So you'll have to calculate the magnification of each scale bar, and I'll just do that for you. Now you can see how I have calculated, 2 centimeter is equal to 20 millimeter is equal to 20,000 micrometer, and you divide it by the 2. Then that is 10,000 magnification. Then 100 nanometers, so 4.5 centimeter divided 4.5, 45 millimeter, 45,000 micrometer, another 3 zeros nanometer. You probably won't be asked in the O levels nanometers. Then 50 micrometer is 2.5 centimeter, measure the length, and measure the length of what? This is what you have to understand. This is the measure the length of this. This is 2.5 centimeter. 25 millimeters, you have 25,000 micrometer, because this is also in micrometer. And then you divide it by the 50. I've written the 50, I've shown it to you as the green. This is the 50 which I'm talking about, and this is the 50 here which I've divided it by. So the magnification is 500 times. So now let's go back to the question and see if we can figure it out. As we know, bacteria, this would be a bacteria. So a bacteria is of course bigger. Then we have a virus, because you can see a protein coat. And this would be a fungus, but it's a fungus spore. And these are of course microscopic. Now you have to see bacteria is the largest, then virus is the tiniest, and then of course the fungus would be larger. So the answer is C. But then please understand why the answer is C, because here on this scale line, we would write 2 micrometer. On this scale line, we would write 0.1 micrometer, because I told you in the question that this is micrometer, and here we would add, say, 80 micrometer. And this is how we would have done this, because knowing it, that this would be bigger and this would be smaller. But if you understand what is a scale line, only then you could be doing this. This is a very difficult question. I agree with somebody suggesting it to me that this was a difficult question. So you have to understand is how we would do this magnification and how we figured this out, because bacteria being bigger than a virus and then a fungus being bigger than even a bacteria. So this was what you had to figure it out. Now a student just emailed me this question and said that they were not able to understand this question, and so that's why I made the video on this question as well. The diagram shows an investigation of blood flow in the veins of the lower arm. Now, first of all, you've got to understand if it's a vein, it is returning blood back to the heart. So it has to be in this direction, because the blood, of course, the human being is this is so if we have to continue, so it's going to go back to the heart, right? So this is the neck of the person and this is the arm, and sort of if you make a drawing of the person. So the vein has to be carrying blood back to the heart. So if this is the vein you've got to understand, right? It's going back. It has to be in this direction. It can't be in any other direction. Now it says a cloth is tightly wrapped around the arm at point Z. So here we have this cloth. So it is preventing the blood to go back. That is what we do when we have to take blood from a patient. We actually do a tie it here, and so that the veins swell up, so that then we can easily take blood from the patient. And please remember, we take blood from the veins, not from the arteries. Arteries have blood at very high pressure. So a cloth is tightly wrapped around the arm at point Z, and the veins stand out clearly. Why is the vein standing out clearly? Because now less blood is returning, so this is becoming thicker and thicker. So they stand out. Stand out means they become more prominent. One finger presses firmly on the vein at W. So we've got one finger here, right? One finger here. And then what have we done? When another finger strokes off the vein, so then we're going to empty the vein like this in this direction. So we are helping the blood to empty out. When another finger strokes the vein as shown in the diagram, the vein lies flat between points W and Y. So you put a pressure here, and then you've emptied the vein, so it lies flat. It is not that, because when you say this stand out clearly when you tied the bandage, now it lies flat, because what have you done is you push the blood to this side and the vein is now empty, right? Let me explain it again. Vein, blood flowing in the veins flowing in this direction. Now you stand. Why do the veins stand out clearly is because you have wrapped this cloth around this place here. So tied a sort of a band around here. So the veins will become more prominent. They become thicker because now the blood is sort of, there's a little restriction. It can't sort of collect here. A little bit is flowing, but there's more accumulating here. So there's a slight backlog. There's a slight back. The vein is becoming now fully distended. So one finger presses firmly on W. So we've got this finger pressing at W. And then what have we done? Another finger strokes the vein as shown in the diagram. The vein lies flat between points W and Y. So now we have emptied it. We have emptied it from here to here. So now it lies flat between W and Y. 4 reasons are given to explain why the vein lies flat. The bandage at Z prevents backflow of blood. Backflow of blood is this way. No, that is not the right way. It doesn't prevent backflow of blood. It is just preventing the blood in the veins from going in that direction. Then the second point is the finger pressed at W prevents more blood entering the vein. Yes, from here it is preventing more blood because you press the vein here, but it will go in the other because the other branches where it will go to. So 2 is correct. Then a valve at Y prevents the backflow of blood. Yes, if there is a valve here, then the valve is something like this. So it is going to prevent the backflow of blood because blood won't go back then. That is what veins have. Veins have valves. So 3 is also correct. Now, why is Z wrong? A valve at Z prevents more blood. A valve at Z, no, you've tied a bandage here. We don't know if there's a valve at Z. Prevents more blood from entering the vein, so this is wrong. So that is why the answer was C, which is 2 and 3. Another challenging question. Seals are mammals, marine mammals. When they dive underwater, they are capable of respiring anaerobically for long periods. We can't do that, but they can. During this time, blood flow to the muscles is greatly reduced, but the muscles are able to tolerate high concentrations of lactic acid. The graph shows the concentrations of lactic acid and oxygen in the blood of a seal before, during a dive, and after a dive. So the 3 things: before, during, and after a dive. Now, period of the dive is this part. This would be before, and this would be after. Now I've given you a picture. This of course wasn't given in the paper, but I want you to see it so that you can visualize it and you can understand it. What explains the change in lactic acid concentration during time X? Now this is time X. Now this is after the dive. Increased lactic acid production, increased blood flow to the muscles, increased rate of aerobic respiration, reduced rate of anaerobic respiration. Now, what is happening during time X? You can see the oxygen is increasing and the lactic acid both are increasing. Now, as you can see, what is happening during, it says explain the changes. What explains the change in lactic acid concentration during the time X? Now, during the time X, if you look at it, the lactic acid is remained the same, and then after time X, it's increasing. Why is lactic acid increasing? Now, what is this? It is concentration in the blood in arbitrary units. Say it was 10, now it's gone up to 50. So if this was 10, now it has gone up to 50. So what you have to understand is how do you explain that the change in lactic acid concentration? Because as the dive has finished, at this time. So there is no more lactic acid is not being produced. So lactic acid production is wrong because the period of the dive has finished. Now the only answer could be increased blood flow to the muscles. Why? Because why are the others wrong? Increased rate of aerobic respiration. Well, the exercise is finished. Why would there be more aerobic respiration? Aerobic respiration releases more energy. Reduced rate of anaerobic. Anaerobic has also stopped because the dive has stopped. You have stopped the dive has finished. So the seal is no longer anymore doing is underwater. Now it's come to the surface. So it can't be D also. So this is the reason why there is increased blood flow, and that is why you're seeing more lactic acid entering the blood. Because lactic acid is produced in the muscles, but it enters the blood. So if there is increased blood flow, only then more blood flowing per unit time, so more lactic acid entering the blood. Of course, and then why is it decreasing? Why is it decreasing? Because more of the lactic acid is taken to the liver, where it is again converted back to glucose, and then the glucose is required to release energy. Another difficult question, the diagram shows some of the structures seen in section through the human skin. What is the function of structure X? Well, if you know structure of function X, but this is of course, this is the sweat gland. So if I label this, what is the function of sweat gland? You know it is when you are exposed to heat. If you enter this room is very hot, then the sweat production starts. So now the point that you've got to remember is there must be a motor neuron supplying this. I said motor neuron. I'm helping you to revise this and maybe a question comes on this. Why? Because motor neurons supply muscle and glands. So here you have this muscle here is a hair erector muscle. So this would also have a motor neuron. Motor neuron means motor muscle or gland. So this would be the hair erector muscle. So they motor neuron. So there would be a motor neuron here as well, which is supplying the hair erector muscle. And of course, the sweat gland must also be supplied by a motor neuron because when you enter this room is very hot. Sweat production starts. Now, how does the sweat gland know when it is got to produce sweat? The motor neuron brings impulses which makes the sweat production, which then the cells of the sweat gland take the water, salts, and urea, and the sweat is formed, and that sweat goes to the surface and then it evaporates from the surface. So production of sweat does not cool you down. It is the evaporation of sweat which cools you down. So that is why the question 22 is, what does it do? What are these structures which they have labeled X? Now these are receptors. And what is this neuron? This is the sensory neuron. And the sensory neuron detects changes in temperature. So you enter this room is very hot. How did you know that? Well, it's the sensory neurons, it's the receptors in the skin which have detected it. Not your eyes, not your nose, not your ears, but it is the skin, the temperature receptors in the skin which detect this change. Information goes to the brain. The brain sends impulses to sweat glands produce sweat, or there is vasodilation and so on. So to cause capillaries to constrict, no. Capillaries, number one, cannot constrict. Capillaries do not have muscles in their wall. To receive impulses from the nose, they don't receive, they take impulses to the central nervous system. To stimulate sweat glands, no, this has nothing to do. Sweat glands will be stimulated by a motor neuron. Question 33, another difficult question. A person drinks a large amount of water. How does this affect the water potentials of the liquids in the renal arteries, the renal vein, and the ureters? Now, what we have to understand is, where does this water go? The water is absorbed in the intestine, it enters the blood. So, all over the body, there is more. So, more in the aorta, more in the arteries. So, water potential in the renal arteries would increase. Why? Because there will be more blood. But then, when the renal artery enters the kidney, if you remember, the renal artery enters the kidney, and here we have the glomerulus. And what do the kidneys do? It removes the excess water. So the renal vein coming out of it is going to have less water, because what has happened is that water and excess, this thing is moved, goes into the ureter. So the renal vein will not change. There will not be a lot of change of water potential. Water potential means more water or less water. More water has the distal water has the highest water potential. When you add a solute, it decreases the water potential. So water potential in the ureters, yes, because more water is being passed out in the ureters, so it will be large volume of dilute urine. So that is why the answer to this is B, and the others will be wrong, because renal vein is the one which is going to have, there isn't going to be much water in that, because the water has been removed by the kidneys, because that's the function of the kidney, to remove excess water, excess salts, and to remove urea. Now question number 36, which row correctly describes the connections of the neurons in a reflex action? Connections of neurons in a reflex action. I've given you a reflex arc at the bottom. Of course, this would not be in the question paper, but I've given it to you. Sensory neurons do what? Sensory neurons carry impulses from the receptors. Then you've got the cell body out here, and then it goes to the spinal cord. And this is the spinal cord, is the central nervous system. So receptor to central nervous system would be the sensory neuron. So the only answers could be then between neurons you have the relay neuron. You can see there's a relay neuron here. And then the motor neuron goes to muscles and glands. So central nervous system to effector. Effector can be either a muscle or a gland. So motor neurons will go to muscles or glands. And please revise this so that everybody is very clear. Just pause the video here and have a look at it. I'm sure you all remember it, but please revise it. Now, another very tough question which you all find a little difficult. The graph shows the relative risk of lung cancer according to the years of smoking and the number of cigarettes smoked each day. Now, if you look at these graphs, first try to understand, okay, does the graph tell me this? Now, what is this telling you? Over 20 cigarettes per day, the risk was less. The risk was 5, 10, 15. So risk was okay till 15. And then after 40 years of smoking, the risk seems to increase. Explain this yourself to the graph. Explain the graph to yourself. What are things? What is the graph telling you? Then the people who were smoking 10 to 19 cigarettes per day, the risk was there. It was increasing. It increased more here after 30 years of smoking, and then it increased even more. This is the years of smoking here. Then the people who were having less than 10 cigarettes per day, their risk increased, but increased after 40, it increased gradually, but then it increased very sharply after 40 years of smoking. After 40 years of smoking, the following statements were made. Now, what you have to understand is when you read this question, you say, does the graph tell me this? Does the graph tell you this? The following statements were made. The relative risk of lung cancer depends on the number of cigarettes smoked each day. The relative risk of lung cancer depends on the number of years of smoking. The relative risk of lung cancer depends upon the tar content of the cigarette. Now, where is the tar content of the cigarette mentioned in the graph? I would have said, when I read this question, this is the first time I read this question, I said, well, this is wrong. So wherever 3 was not applicable, there was a very simple way of doing it. Which statements are supported by the data in the graph? And there is only one here, in which 3 is the one which is the BS, beautiful story, not something else which you are thinking. So the 3 was wrong. Why? Because the graph does not tell you anything about the tar content. There's no such mention of the tar content. What is only mentioned is the relative risk, the number of cigarettes, and the years of smoking. That's all what is given to you. So I would have figured this out with a simple way of answering it, and this is how I would have figured it out. So whenever you have such a question, you've got to ask yourself, does the graph tell me this? Does the graph tell you that the relative risk of lung cancer depends on the number of cigarettes smoked each day? Yes, there's some people 10 cigarettes, 19, 20 cigarettes. So this is telling you that. Then the relative risk of lung cancer depends upon the years. Yes, the years of smoking is also given here. So the years of smoking is also given here. So this is what the graph was telling you, these things. And the graph is not telling you anything about the tar content. So there is no mention of the tar content of the cigarette. So why are you thinking that? So that is why you've got to understand is the answer was B. And this is the last question that I have done. I will try to upload another video with the difficult questions, and I wish you all the best of luck for your exam day after tomorrow. And if you have any other special questions which you all would like to ask me, please do refer them to me, email them to me, and I'll have a look at it. Thank you very much. Thank you for subscribing. Thank you for watching the videos.